this week we'll be looking at sample problems for week 11's lecture. The first question is the principle of amplification of DNA with a modified nucleotide used in sequencing of biomolecules is by what method? And four different methods are given here: Sanger method, Maxam-Gilbert method, Edmond degradation, and mass spectroscopy method. So, like as always, you can begin with mentioning an incorrect option and justify why an option is incorrect. You can take a couple of minutes to think about this and then we'll discuss. Yeah, so you can start with an incorrect option. Does anyone want to try answering?
Sir, mass spectroscopy method is a wrong answer.
Okay, what do you know anything about that method?
It is not for use for sequencing.
Sequencing of what?
Sir, mass spectroscopy is used for quantification,
Quantification of what?
like nucleic acids, proteins.
So what do you measure in mass spectro? So it's actually mass spectrometry. What is given here is it should be called as mass spectrometry, not mass spectroscopy. That's okay, but what is quantified there? What do you measure there?
Determine the molecular say, weight of the nucleic acids or protein.
Right. So generally, so in mass spectrometry, what you do is you have your biomolecule of interest that is put on a matrix, and then you shoot a laser at it. The thing is, it gains energy, and in an electric field, it is allowed to move. So what happens is the distance that it is supposed to move is fixed. The time that it takes will depend on how heavy the molecule is. Based on that, we will be able to derive the mass. So with this, you had mentioned that it's used to quantify the molecular mass. But can you do sequencing with mass spectrometry?
No, sir.
Okay, people actually do that. That's a routine technique. So what people do is, let's say you take a protein. It's not very well established for nucleic acids because you have other better methods. But for proteins, what people do is they take the protein, they digest it with random proteases. So the cleavage is not really known. So once the digested samples are put on this matrix, they can fly. This is calculated as the time of flight. Based on this, they back calculate the mass, and according to the mass, they try to tell that this is the probable sequence. This is how protein sequencing can be done using mass spectrometry. So you can just make a note of that, that you can do protein sequencing using mass spectrometry. Right. Other options? Does anyone want to answer? Nityashri, Subramanian. Okay, so among these, Edmond degradation is another method where you have sequencing of proteins but not nucleotides. Maxim Gilbert method and Sanger method, these are the methods that are used for sequencing of DNA. While one method uses dideoxynucleotide, the other method uses uh, piperazine. So we'll come back to these later, but just a brief overview. So in Edmond degradation, what happens is you take your protein, you put it with phenyl isothiocyanate. So now this is added to the uh, uh, first residue, that is the N terminal, which has the NH2 group. Then it is cleaved with thiazolinone. Now, because of this, now you have one amino acid. Now, then this is converted and based on chromatography. So, let's say you have a particular amino acid that is covalently modified with this phenyl isothiocyanate, and if you run it on HPLC, you will know its retention time. Based on that, you will for every amino acid that is being broken down and separated, if you run it, you will be able to get the corresponding retention time with which you can accurately tell what is the amino acid there. Yes. Is there a question? That's what I told. Edmond degradation is used for sequencing of proteins. That's what I've been explaining here. You can unmute, Subramanian. Hello. Yeah, Sai Sampath, was it clear? I mentioned that this is done for proteins.
Yes, sir.
Okay, fine. Yeah. Uh, Yeah, so the other method that we have is in Sanger method, what we do is we use dideoxynucleotide. So here your chain is terminated, and based on chain termination, people uh, identify this. Now that is what is being asked here. A modified nucleotide. We have the structure of it later, so we'll go to that. So in this method, in Sanger method, the nucleotide that will terminate the reaction by not allowing the next nucleotide to be covalently linked is added. So, so four different reactions are done. In each of this, you have uh, all the four deoxynucleotides and one of the other. So, for example, in this case where A termination is there, you will have DATP, DCTP, DGTP, and DTTP. And if you have DATP, what will happen is at different positions it will terminate. And when you run this on a gel, you will get bands accordingly. So based on this, we will be able to get a sequence. Okay. The next is a relevant question. Structure of DDNTP, and four options are given here. It has both 2' and 3' OH. It has 2' OH group. Option C, it lacks both 2' and 3' OH group. Option D, it lacks 2' OH group. So you can take a couple of minutes to think about this. So before you answer, you can think about how in a particular in a reaction where this DNA polymerization is happening or DNA synthesis is happening, uh, where is the bond being formed? You can first think about this, and you can also define the structure of DNTPs. Based on that, you will be able to answer this. You can think for a couple of minutes. Okay, so does anyone remember uh, where the covalent attachment of the next nucleotide happens with the previous nucleotide in a DNTP?
Sir, at a normal sequence, 3' OH to 5' phosphate, it forms phosphodiester bond, 5' phosphate.
Yes. So that means in a DDNTP where we want to terminate the reaction, definitely 3' OH cannot be there.
It lacks 3' OH, sir.
Yes.
DDNP.
DDNTP, yeah. And should it lack 2' OH as well?
I have no idea about that. I know about only 3' OH it lacks.
Okay, so structurally, if you think about this, there is a reason why it's called deoxy, right? So this is a nucleotide. Okay, so uh, you have the 2 prime and the 3 prime. So now in this nucleotide, if you remove one OH, that is deoxy, that is your DNTP. In DDNTP, it is dideoxy. So two of these OH are removed. So both of these are not there, which is why there is termination.
So why to terminate 2 prime with?
No, no. So that is naturally present. So what I show you here is a nucleotide. Usually, okay, the name deoxy ribonucleotide, it comes from the removal of 2 prime OH. So in those nucleotides where you do not have 2 prime OH, those are called deoxynucleotides. We don't say the same for RNA because in RNA you have both. Was that clear? Or I'll repeat.
Yes, sir. I understood, sir.
You understood. Okay. So in RNA we have both 2 prime and 3 prime. Of which 3 prime will help in your covalent linkage. But it when you remove the 2 prime, it is called as deoxy. And when both 2 prime and 3 prime are removed, it is dideoxy. Right. The next question is Klenow fragment lacks dash polymerase activity, 5 prime to 3 prime exonuclease activity, 3 prime to 5 prime exonuclease activity, proofreading activity. So again, you can take a couple of minutes to think about this and then start with uh, where this Klenow fragment is derived from and how large it is, like whatever information you know about this. Maybe you can start with what you know about DNA polymerase. We can build it from there and come to Klenow fragment specifically. DNA. First, we can start with what we know about DNA
Okay.
polymerase. We'll take it from there. We'll build it from there. Yeah, you can start
Uh,
with what you know about DNA polymerase.
Basically, it is DNA polymerase 1. It has 3 activities: 5 prime to 3 prime exonuclease activity, 5 prime to 3 prime polymerase activity, and proofreading activity.
Okay.
So once the protease can cleave the DNA polymer 1 to 2 fragments. So that one of the fragment is Klenow fragment, which has 5 prime to 3 prime polymerase activity and a proofreading activity, and it lacks. And another fragment has the 5 prime to 3 prime nucleus activity.
Right. Yeah. Do you know what exonuclease activity means? And do you know the difference between exo and endo?
Exo means, it means cut the part of the DNA, right, sir?
Yeah,
In the 5 prime to 3 prime.
nucleases cleave DNA and RNA. Ribonucleases cleave specifically RNA. Nucleases, deoxyribonucleases cleave DNA. Right. Now, do you know the difference between exo and endo?
I know about exonuclease, endonuclease. I don't have much idea.
No, no. You
Forgot it.
okay? No, no. I'll give you a clue. You can try thinking. Have you heard of restriction enzymes?
Yes, RE1, RE2, like I remember.
Yeah. So where does restriction enzymes cut?
At the restriction sites.
Yeah,
Specific sites.
Specific sites. So exonucleases will require something like a 5 prime phosphate, which is present only at one end, which is why it can cleave from 5 prime to 3 prime, something of that sort. or it requires a 3' OH. That is how it identifies. So it cleaves from either end of the strands, depending on the orientation that is given here, 5' to 3' or 3' to 5'. Whereas endonucleases are the ones that you, like you may have mentioned, it cuts within a DNA strand. It doesn't require any of these 5' phosphate or 3' OH for recognizing and cutting.
Sir, restriction enzymes are part of endonuclease, right, sir?
Yes, restriction enzymes can be classified into endonucleases.
Yeah.
Yes, most of the restriction enzymes like EcoR1, which is what people generally read about, so that can come under endonuclease because it has a specific site GAATTC which it cuts in between. Right, good. So, like you had mentioned, so the DNA polymerase is a very large enzyme which has three activities. One is for the synthesis, that is 5' to 3' polymerase activity. Then it has this exonuclease activity, and it has two different exonuclease activity. One is 3' to 5' exonuclease activity, the other is 5' to 3' exonuclease activity. So in this 3' to 5' exonuclease activity is where we have the proofreading. So when we cleave this with a protease, what happens? There is this other fragment which is needed for removing the primer. That is why you need the 5' to 3' exonuclease activity. Now, when you cleave this with the protease, this is lost because these are different domains that are linked by a small set of amino acids, and that is lost. So, in that time, only this 5' to 3' polymerase activity and 3' to 5' exonuclease activity, which is needed for proofreading, is retained. That's what even Sai Sampath had mentioned. Good. The next question is, in the presence of NaCl, hydrazine will modify dash base in the DNA. Cytosine, pyrimidine, purine, and guanine. So you can take a couple of minutes to think about this, and then we'll start with, maybe if you know about the technique, we'll start with what the technique is about, and build it from there. Okay, so this method where hydrazine is used, that will modify bases in the DNA. So this comes under Maxim Gilbert sequencing. So here what we do is they take the single strand DNA, then involve piperidine, and then they take chemicals that can selectively or specifically act on purines or pyrimidines. We cannot really tell whether it acted on an AT or GC, but based on getting results from combination of G plus C and G or C, we'll get to know which base is in a particular position. So the idea here is purines, it will react with dimethyl sulfate, and pyridines will react with hydrazine. That way, uh, your glycosidic bond is broken. Then this piperidine, this will catalyze uh, phosphodiester bond cleavage. Now, when dimethyl sulfate and piperidine act, then guanine will be cleaved. And in formic acid, both guanine and adenine nucleotides will be cleaved. If we replace dimethyl sulfate with hydrazine, what will happen is it will cleave both thymine and cytosine. But when we add NaCl, only cytosine is cleaved. We have a picture of this later. Now, with this, we'll be able to uh, uh, so when we do all of these combinations and run it on a gel, based on the number of bands and the gaps, we'll be able to tell in a similar fashion to how we get it from this dideoxy termination method. We'll be able to recalculate and tell what the sequence of our DNA is. Now the question is, in the presence of NaCl, hydrazine will modify dash base. So like it is given here, hydrazine will specifically act on pyrimidines, that is cytosine and thymine. Like it's given here, when 1.5 molar NaCl is added, it will cleave only cytosine and not thymine. So this is the specificity that was asked. Fine. A similar question on the same lines. In Maxim Gilbert sequencing method, dash catalyzes the cleavage of phosphodiester bond next to the modified nucleotide. So we earlier mentioned about a brief overview about Maxim Gilbert sequencing, where dimethyl sulfate and hydrazine are used. Now these can specifically act on, I'm just repeating what I had previously told, these can specifically act on selective bases, and piperidine will cleave the phosphodiester bond that is next to the modified nucleotide. So among these, the right answer is piperidine. One more question along the same line. Now, this is about identifying the sequence. So, in Maxim Gilbert sequencing method, a band was observed in both G plus A and G lane in the gel. The corresponding base in the DNA sample is dash A, C, G, and T. Now, because we have a band at both G plus A and G, this would mean it is G. That's a commonality. That's how we identify. This is again a depiction of how this happens. Okay. The next question is the role of carboxymethylcysteine during sequencing of protein is to dash, and four options are given here: to allow formation of disulfide bonds, to prevent formation of disulfide bond, to allow refolding of protein, to oxidize the cysteine residues in the protein. Okay. So you can take a couple of minutes and think about what cysteine is and what role cysteine plays. Based on that, we'll take it from there.
Sir, cysteine is a sulfur-containing amino acid,
Right.
And it's a building block of protein, sir.
Yeah, every amino acid is the building block of protein. That is right. Do you know any other amino acid that has sulfur other than cysteine?
Methionine, sir.
Right. Okay. So, what is a specific feature of cysteine that contributes some structural stability to proteins? By structural feature, what I mean is you need energy to break it, and it uh, ensures the protein is more stable. Is there any kind of specific bond that cysteine is usually observed to form?
The cysteine can form disulfide bonds.
Yes. So disulfide bond is a covalent bond, unlike other bonds that we read about, non-covalent bonds like hydrogen bond and so on. So because disulfide bonds are covalent, it restricts amino acids conformationally, thereby giving it rigidity and stability. Now, what is mentioned here is carboxymethyl cysteine. That is a modified cysteine. So, okay, there's an issue with one of the pictures here. It, okay, sorry. I had pasted one as carboxymethyl cysteine, the other is normal cysteine. So what you see here, both are uh, carboxymethyl cysteine. So usually in cysteine, what you have is you give me a minute. I'll help you understand it. Uh, okay, let's say. Okay. So usually in cysteine, you have this an SH group. Okay. But in carboxymethyl cysteine, you have a CH3, I mean CH2, and then COOH. Uh, Now, when you have an SH group, it can form a disulfide bond when another SH group comes close by. But when the valency of sulfur is being satisfied here with this carboxymethyl group, it cannot anymore form a disulfide bond. So that property is used during sequencing of protein. So what they do is uh, they chemically modify one batch and do not chemically modify the other batch. So it's like you have a wild type protein, and in the other batch you have a protein where it is modified. And when they digest it, the time of flight will be different for both, because one of it contains a disulfide bond, the other doesn't. And based on degradation and time of flight, they'll be able to tell that this amino acid forms a disulfide bond with this. So like we saw here, this uh, because sulfur's valency has been satisfied here, it can no longer form a disulfide bond. So it prevents the formation of disulfide bond. That's the required answer for this question. Was that clear?
Yes, sir.
Okay. Okay. The next question is cysteine. Okay. This is slightly different. Cysteine residues in protein are oxidized by dash. Wait, I don't know if they're asking about cysteine or cysteine, but yeah, let me check. Okay, you can take a couple of minutes to think about this and then tell me if you know the difference between cysteine and cysteine. Okay. So if anyone knows the difference between cysteine and cysteine, you can unmute and let me know. Okay, if not, it's fine. So what you see here is a cysteine. When it forms a disulfide bond with another cysteine, that is called a cysteine. Now, cysteine residues, these have an SS double bond. Okay. So now the question is, these are oxidized by dash. So dithiothretol and beta mercaptoethanol, these are all reducing agents. Okay. So reducing agents, what they do is they ensure a cysteine, which is this SH, remains as an SH instead of forming another disulfide bond with some random cysteine. Unless, uh, so usually what we do is when we purify proteins, we add one of these two, beta mercaptoethanol or dithiothretol. We have another thing called TSEP. So all these three are reducing agents that ensure that as you purify your protein, two proteins with exposed cysteine do not come close together and form a disulfide bond. In that case, the disulfide bond that is being formed is intermolecular between two molecules of protein rather than two amino acids within the same protein. So usually disulfide bonds are formed within intramolecular cysteines between the cysteines in the same protein. When you have cysteines from two different molecules of protein, what it leads to is called as an aggregation event that ensures that your protein is not folded the way it is supposed to fold. So to prevent that, we add dithiothretol or beta mercaptoethanol or TSEP. Each of these have different half-lives at different pH and temperature. TSEP being the most stable one. Uh, Other than this, we have something called as iodoacetate. What this does is it chemically modifies the sulfhydryl group, which is the SH group of cysteine. So when this is chemically modified, uh, if this particular cysteine is needed for functionality of a protein, then that cannot happen. So when people use iodoacetate is, let's say you have a protein whose active site has a cysteine group, an SH group, and this is important for the catalysis of the enzyme. Now, when you add iodoacetate, what you do is you essentially make it inactive by adding a bulkier group onto the sulfhydryl group. So, this can no longer participate in the catalysis and hence the enzyme is inactive. And the last option is performic acid. This is a oxidizing agent. So, what this does is it can oxidize the disulfide bond to yield cystic acid. Instead of a sulfhydryl SH, you have SO3H. Right. So these are the four different chemicals, and I hope you understood the differences between cysteine and cysteine. What you see here. Okay. The next question is trypsin cleave protein at dash. N-terminal side of lysine arginine, N-terminal side of tryptophan tyrosine phenylalanine, C-terminal side of tryptophan tyrosine phenylalanine, and C-terminal side of lysine or arginine. You can take a couple of minutes to think about this and then we'll discuss this. Okay. Does anyone know what trypsin is?
So, trypsin is a digestive enzyme, sir.
Right. So it can cleave. Okay, it's a digestive enzyme. What does it cleave? What does what can it digest?
It digests protein, sir.
Right. Good. Yeah. So trypsin can cleave proteins. Now, do you know anything other than this? Like where it cleaves? Can it digest from at a specific sequence at the N term, at the C term, something of that sort?
No, sir.
Okay. So trypsin is specific to positively charged amino acids, which is lysine or arginine, and it cleaves after that amino acid ends. That is why it's written as C-terminal side of lysine or arginine. So if you see, uh, you have this your polypeptide where you have lysine and arginine. So what it does is it cleaves at the 3' end, what you see here. So this is our amino acid of interest. So once the amino acid is getting over, so how we read amino acids is the N terminal is where you have the NH. C terminal is where you have the COOH. When a peptide bond is being formed, you have CO double bond NH. So this is the end of this amino acid. So once this amino acid ends, it will be clean. So you get a CO, a carboxylate ion here. This is how trypsin acts. The next question is carboxypeptidase. So carboxypeptidase will remove amino acids in protein from dash C terminal, N terminal, both C terminal and N terminal, middle portion of the protein. Okay, so carboxypeptidase, as you see from the name, it acts at the carboxy terminal or the C terminal. So when you have an amino acid where this is getting reduced, it removes the n minus 1 amino acid. So, like we said, this amino acid R prime, it starts at NH here. So, this peptide bond will be cleaved. The next question is, the location of disulfide bond in the protein can be identified by dash. Sequencing with or without DFNB, sequencing with or without performic acid, sequencing with or without trypsin, sequencing with or without acid hydrolysis of N-label protein chain. So we discussed about this in a couple of questions back. You can think about this, and you can think what a chemical can modify a disulfide bond and how can it modify. So based on that, we will discuss this question. You can take a couple of minutes to think about this.
Sir, in the previous question, you said like performic acid
Yes.
oxidize the system, the disulfide bond.
Disulfide bonds, yes. So when you have this disulfide bond, it will oxidize and it will give you two cysteines. Huh. So we I had also mentioned about how that can be used in sequencing, if you remember. Was that clear? Or can you try explaining that? Yeah. So do you remember how the location of disulfide bond can be identified? I had mentioned it. Okay, there's no issue. So what people do is they'll take one sample that is not at all modified. So there your disulfide bond is present. In the other case, they'll treat it with this performic acid, which will oxidize the cysteine into cystic acid. This is actually cysteine. It has an E here. Okay. So now each both of these are different because one is the wild type which has a which if you cleave it with a particular protease will give you this covalently linked sequence, whereas in the other case you will get different set of sequences. So to understand this better, have any of you done digestion of DNA with enzymes, restriction enzymes? Yeah, can you hear me?
Yes, sir.
Yeah, have you done digestion of DNA with restriction enzymes?
Yes, sir.
Yeah.
Like in first year, I don't remember, but I have an idea.
Okay, you have an idea. So here also it's similar. Let's say, okay, what enzyme did you use? Do you remember by any chance?
No, sir. This
Okay,
is something I don't remember.
okay. Do you know any enzymes? Any restriction enzymes? Name of any restriction enzymes? Okay, fine, no issues. Let's say you have a restriction enzyme called as EcoR1, the other enzyme called NDE1. Now you take a plasmid and you cut this with NDE1 and EcoR1. The plasmid is circular, so you'll get two fragments. Now let's say we mutate the site where EcoR1 will cut, so that EcoR1 cannot cut. So now what we do is when we run these two on gel, the one where there is no mutation, you will get two bands, whereas in the one where you have mutation, you will see a single band. This is also similar. When we run the wild type protein, which is equivalent to the non mutated DNA, you get a particular set of bands or particular set of time of flight which you can correlate with the sequences. And when you treat it with performic acid, you get differences. So based on these differences, we'll be able to pinpoint what are the amino acids that are involved in this disulfide bond formation. Fine. The next question is, protein sequencing by Edmond degradation is carried out by degrading the peptide from the dash C terminal, both C and N terminal, central amino acid of the peptide. Yeah, so you can take a couple of minutes to think about this. We had discussed this in the first part of the question. I mean, first question. So, to start with, you can tell me what chemical is used to modify uh, or is majorly used in Edmond degradation. We'll take it from there. Okay, so we looked at Edmund degradation in question number one. So here we use phenyl isothiocyanate. So what it does is it adds to the N terminal of the peptide. Then when we cleave it with thiazolinone, uh, we get the first amino acid. Then the second amino acid is now, now you have a mixture of one amino acid as a combination of phenyl isothiocyanate derivative and the other part of the protein. Now, when you run this on a HPLC, based on standards, you will be able to tell which of these, which amino acid is there in the first position. Then you repeat it for the other set of other parts of the protein, one by one. So, here the degradation is carried by degrading the peptide from the N terminal. Okay. Okay. The next question is, for northern transfer, deionized distilled water is treated with DEPC before using it for preparation of agarose gel to dash. So, does anyone know what northern transfer is? Nityashri Sai Sampath?
Sir, we to detect RNA.
Okay. So it will tell you what are the RNA that is being bound to this. If you have a probe specific to an RNA sequence, you can detect that using northern transfer. Fine. Now, do you know what DEPC is?
Sir, I know that DEP is used for killing the RNAs.
Okay. Does it remove RNAs or does it inactivate RNAs?
Inactivate.
Yeah. How do you know how it inactivates RNAs?
No, sir.
Okay. So DEPC is called as diethyl pyrocarbonate. So this is just a representation for Western blot. So in DEPC, what DEPC does is it can covalently link to histidine or lysine, which is usually present at the active site of RNases. So when you start your experiment with DEPC water, you will ensure that any enzyme with an active site, histidine or lysine, will be inactive. So in that water, you will not have nucleases that are enzymatically active. If you add this to fresh RNAs, that will not work because the DEPC would have been activated uh, and it would have modified your RNAs that were existing in your solution earlier. Fine. Two more questions. Which feature of eukaryotic mRNA can be exploited for cDNA synthesis? And four options are given here. Cap of 7-methyl guanosine at 5' end, 5' UTR. Poly A tail at 3' end, 3' UTR. You can take a couple of minutes and then tell me what are the differences that you know between prokaryotic and eukaryotic mRNA. And then we'll take it from there.
So prokaryote mRNA doesn't consist of introns, and eukaryote consists of introns. After the post transcription modification, the introns gets cleared.
Okay. What is the post transcription modification that you're talking about? Is there something specific? Does it have a name?
Yes, at 3' and it forms poly A. Formation of adenine, so like long chain of adenine
Okay.
at a 3'.
No, no. I wanted to know about this post transcriptional modification that you meant for introns and exons. You were about to mention something, were you?
Yeah, I mean splicing.
Splicing, yeah, good. Yeah, so the major differences is like you had mentioned, uh, both of it will have UTRs. UTRs are untranscribed regions. Uh, mRNA from eukaryotes have this guanosine cap, and here you will have both introns and exons, which will be spliced to give further down another mRNA. And unlike prokaryotic mRNA, eukaryotic mRNA has a poly A tail. All of this were mentioned. Good. Now, to make cADNA, what we do is we synthesize a poly T primer that will bind to this, and then you use a reverse transcription enzyme that will start transcribing from this end. So we use this feature of eukaryotic mRNA, which is this poly A tail, by having a primer with poly T, poly DT, and then we just transcribe. That is what is the feature that is being exploited. The last question is terminal transferase enzyme will label dash 5' end of DNA, 3' end of DNA, both ends of DNA throughout the length of DNA.
Sir, in terminal transferase, deoxynucleotides are added to the 3' end of DNA molecules.
Okay, what is it? What is a terminal transferase? Why is it needed? And how is it different from a polymerase?
So terminal transferase is independent, template independent. It doesn't require any template to form the to add the nucleotide.
Okay, template or primer?
Template, sir. I think template, sir.
Right. So for any particular template, it will still be able to add it. That's what you mean, right? Yeah. So what is given here is one such representation. So first we have the eukaryotic mRNA, then we have the other primer with poly T. We'll first amplify this. Then we have a terminal transferase that will attach the other primer here. So it can directly add to this. Yeah. So, so with this we come to an end for today's set of questions. If you have any specific question, I'll stay on the call. You can ask me.
Sir,
Yeah. Yes.
sir, I have applied to the NPTEL exam, sir.
Okay.
And I paid 1,000 rupees money, sir. But in payment status, I will check that payment is not yet, sir. It's not opening, sir.
I am not sure about this, but I think you can still email them. You can email the